I'm going to introduce you.
Yes.
So everybody, not that you need an introduction. Good to see you, though. I'm seeing you after a long time. So our speaker today is Professor Luca Giuggioli from University of Bristol. Luca's talk will be on territorial random walkers and the collective emergence of animal territorial patterns. Just to give you a little bit of background, Luca and Nitant Kenkre, who is here, they have a book coming out, Theory of the Spread of Epidemics and Movement Ecology of Animals, an interdisciplinary approach using methodologies from physics and mathematics from Cambridge University Press to appear in December 2020. So this is this is a what is it called? A book release kind of a thing, I guess. No, I'm kidding. So Luca did his PhD in physics at the University of New Mexico with Professor Nitant Kenkre in 2004. He did a postdoc at UNM and then took a postdoc with Simon Levin in the Ecology of Evolution group at Princeton to learn as much as possible about animal ecology. He subsequently moved to the University of Bristol, where he is an associate professor working on applications of statistical physics to ecological problems. His keen interest in ecology made him join forces with an ecologist to found the interdisciplinary journal Movement Ecology in 2013, of which he is the co-editor-in-chief since 2013. His areas of work include collective movement models and more broadly, statistical physics of transport processes and their applications to ecological problems. Lately, he has been particularly involved with the study of dynamics of non-Markov Brownian walks and with lattice random walks in finite domains. So with that, and my apologies for being late, let's get rolling.
Okay, thanks, Surajit. Thank you very much, everybody. Okay, so let's start. Okay, no, wait, so do I do two? Yeah, I just have to click on it. So just a little outline of what I'm going to talk to you about. So I'll just give you a little brief introduction about territorial behavior so that we know exactly what we are talking about. And so then I'll tell you about this territorial random walk model, this collective movement model that we have built. Then we'll analyze some of these phase transitions that appear in this territorial random walk model, and in specific, this will be about scent marking animals, so those that are territorial via scent marking. And then we'll talk about some analytic approximation. I'll give you the detail of the one-dimensional model. I'm not going to show you the horrible expression in 2D, but those are the ones that have been used to actually fit the data in a fox population. I'll tell you more about that. And it was an episodic of Ming back in the 90s here in Bristol. And then probably just a very brief conclusion. So let me start with introducing a bit about the biology of territoriality. So what is considered animal territoriality for biologists? Well, it's very simple. It's a result of a behavior that has as a purpose the need to exclude from certain region of space what sometimes are called conspecific. So animal of the same population or same species, if you want. So now, what are the mechanisms with which they do that? Well, it depends on the senses they use to do that. It's also called territorial defense. You know, when you have territorial behavior, you defend the territory. It's called the territorial defense. It's sort of a synonym.
What is conspecifics, Luca? I'm sorry.
Species, individual of the same species.
Okay, I'm sorry. Okay.
Nitant is smiling because he hates that word.
Yes, you got that question.
So then the mechanism with which it happens, it depends indeed on the species. And then you can have, you know, as you can see a baboon here, they use, you know, audio signals to actually make sure that individuals know that they are there. Then you can have some visual displays. That bird, I'm actually not sure which one is that. But, and then you can have actually scenting. So these are olfactory signal or cues, and they are deposited, and by typically red foxes, many of the canids, they do that. So, and now in physics, I'm going to argue that, and this is what this talk is about, is to show you that indeed one has to deal with this problem as a collective movement model, a collective process, sorry, where territories move continuously in space and time as a result of the wandering of the animals that underline it, construct these territories, whichever, if you want, mechanisms they use. And so we'll worry about olfactory cues because I'll tell you about scent marking territoriality, but in general, it's valid in general. So, oops, there is a line that I'm not sure why it's there. So let's see the different kinds of species, what they do. Indeed, the one that I showed you with the two pictures, the first two, which is auditory and visual signals. And those, differently from the scent marking ones, they require animals to be in sort of relative proximity. Not necessarily in contact, but relative proximity. So you need line of sight, or you need to be close enough to hear the sound. Now for the foxes, which is an example of when olfactory cues are used, then you don't need that. In fact, you'd say that they use a broadcasting mechanism, since whoever tune in into that broadcast means that they've gone to the location where someone else have left a scent, and so they recognize someone else have been there, and they respond to that. So the first two cases are called direct interaction, since they have to be there when the other individual signals their presence. The olfactory cues, instead, we talk about indirect interaction because animals just leave a scent and they can just go on their life, and then that's it. So that only who goes in that location find out that the scent was there. That's why they interact, therefore, indirectly through the environment. So, in fact, there is a name in biology for those kind of interaction. There is a special name which is called stigmergy, when the interactions are mediated by the environment. That's a name that's been used, has been coined in the 60s to actually look at how termites build their nest. Since, you can imagine you don't need any memory, you can be really not a very intelligent animal in as much as you respond to what you see in the environment. The environment has the memory, and you are genetically imprinted to do certain behavior out of that. And so that's how these nests are built. So this is sort of collective intelligence of, insect that probably you've heard many times talked about. So now, in terms of questions that researchers try to answer on territorial studies, what they do, well, I would just distinguish them in the usual way. You have the why question, you have the how question, and you have the when and where. So when and where you measure this territory, if you want size, etc., and other quantities. So the why questions are the one where you are interested to know precisely the reason behind this behavior, territorial behavior. And clearly you can think about access to resources. It could be mates, food, shelter. In some cases, it's not even that. It could be just a social status. And so, so these are the why questions. The how question is the one that we are interested, which are what are the mechanisms for which these territories emerge, and how are they maintained also. And the when and where is, it's what are the best convenient way or methodology that you want to measure this territorial behavior, and what are the outputs of this territorial behavior that you can actually measure. So the sort of why question typically are what you might call optimality question. These are evolutionary kind of biology questions. What's the advantage for the species, not just the individual? What's the value to the fitness of the individual? These are the sort of questions that evolutionary biologists tend to ask. The how question are the questions that probably physicists would tend to ask, and we did. And so these are what I would call the interaction question. And then the when and where to measure, I call it home range question, because as you will see when I define what a home range, different what territory is indeed when you go and measure some size of these territories in one way or another. So we'll worry about the interaction question then. Now, so when we talk about, as I said, we are talking about scent marking animals. So if you have a formation of a scented territory, therefore, so now the territory is generated by the scent marking, that's why I call it scented territories. There are three processes, if you want, involved that you have to think about. You have a scent marking process, so someone has to deposit a scent. Then you have to defend that territory, which means that someone has to respond to that presence of the scent in the environment. And then the result is that you want to avoid the location where someone else has deposited the scent. No? So if you want, you have a scent deposition, then you have the important aspect is that the scent is a short-term cue, so it stays there only for a finite amount of time. It's not there forever. So if you need to go and to defend your territory, you need to go and defend the territory. So you have to go and renew the scent in the location you have been in the past. And then that's a response of the others, so that the territorial system is stable, if you want, is that everybody retreats upon a country of foreign scent. So this system is sort of there to avoid individuals to always get into, highly costly conflicts, and so maintain a sort of, everybody's happy, to maintain their own areas, of importance. And so that's what you would call it scent-mediated interaction. And if you want, since I explained to you the name stigmergy, that's also a stigmergic interaction. Okay, so now, if you want to model scent marking territorial behavior, or sorry for that, already there. So you need to think about agents that move randomly on a lattice just for convenience, and interact through some sort of spatial exclusion. And that exclusion happens because whenever you find a scent which is active, as I said, the scent is a short-term cue, you retreat. And at the same time, you individual, you keep leaving, scent wherever you go, so the others retreat from where you have been, and so that you maintain certain area of exclusive access. So now, as you can imagine, as physicists, how do we model these agents? Clearly as random walkers. So they move a random walker, if you want to make it as simple as possible. And then you keep a track of wherever they go. So there is a site, to each site which is visited, you have an associated mark, which is on and off. So think about just a binary value now for the moment. And depending on the species, you can have animals that actually mark everywhere they go, like the foxes in Bristol. So these are called hinterland markers, and there are others that only marks the boundaries. And so these are called borderland markers, like hyena, apparently brown hyena. That's what they do, because they deposit scent which lasts much longer and is much more costly. So foxes, on the other hand, they deposit urine, so it's much easier to do. And now the scent, as I said, it decays over time, and so the important is to understand how does it decay over time. So that's an important part of the model. And then again, the retreat is sort of, well, you can also have some liberty on how you implement that, but you can just think about you go back where you came from. So you avoid others to come into a foreign territory. Okay, so since I mentioned about the scent decays, so to simplify the situation of decays, you can think about
Luca, your volume went down, I think.
Oh.
Yeah, there we go. Good.
I'm back on it?
Yeah. Thanks.
So the decay dynamics is an important part of the model, and what's the functional dependence of that decays is very much a biological interesting question, but of course we need to make some choice, make some approximation. So, but certainly yeah.
You mentioned conspecifics, but you might imagine that any two individuals that are competing for a given resource, even if they aren't conspecifics, let's say they have the same prey or something, could also have this territoriality, even though it won't be the same as when they're also competing for mates or something. So do you consider that class of problems as well? No,
yeah, absolutely true. That's correct. I haven't considered that kind of problems, but you're right. So in reality, just to add to what you're asking, is that animals from scent, they can determine so much about whoever has left that scent. So right. Which can be the sex, the size, the age, all sorts of things. And clearly, if there is a competitor, probably even more, they are more even concerned about that. So yes, you're right. So, but I haven't studied a specific kind of problem like that. Is that, yeah. Nice. So since I was trying to say the decay dynamics of the deposit sense is an important part of the model, and you'll see we have made some specific choice, but we have also studied some variation to it. So I just mentioned that if you imagine that P of tau is this probability of retreat, depending on the age of the scent. So whenever you go to a certain location, then that scent has been there for some time. And then you can, these are simple choice of the functional dependence that we have put in to just study what happens to the emerging occupation probability of where the animals go. And then clearly you can see if you have the black curve, which is nearly like a step function, then as you can see, I just pick out two animals out of the entire population, then the overlap of what is that called the home range. I'll define more precisely what the home range is later. So that's very minimal. And then if you pick the blue one, then of course you have lots of overlap. So just to simplify things, what we're going to tell you about, I'm going to tell you about on the case where you have a step function. So it's a binary value. So either the scent is present or is not. And therefore that means that the neighbors at each moment in time can only overlap at the borders. So now, then I'll tell you what home range, so not to get confused, because home range, you can have overlap, which is different. But let's come back to this, with this choice of the probability of retreat upon the scent age to be that step function. And now we can talk about what the territorial random walk model is, at least the original version. So we consider this retreat, which is a step function. Then how do we implement spatial exclusion? So just to give you, I give you a snapshot of a simulation of this lattice. You have two animals there, I have a red and the blue. So so let's say, suppose it's discrete time. Now you suppose you have to move the next step. Now they are random walkers. The red one is not in a location where there is a blue scent. So it's in a normal state. So it chooses either of the four directions in that lattice. On the other hand, notice the interaction happens on the blue because previous step came into this location where there is still an active scent from the red fellow. And now the next step is going to retreat. That's how avoidance, the exclusion happens. And you can implement in different ways, but you can just say either you retreat back where you came from, or you can just retreat randomly wherever there is no red scent, for example. So not much is changing if you do those choices. So essentially that's what the territorial random walk model is. Very simple, the construction. So you have lots of these animals. They get excluded by finding the foreign scent, and the foreign scent is either present or absent, depending on the probability tau that I've just plotted above. And that decay is a given parameter of the model, which is a TA for T active scent. So at the end of the day, so the parameter in the model, of course, as you can imagine, it will be the population density. And if you want to change the movement statistics, which we have done, I'm not going to tell you much about it, but if you configure correlated walkers and so on, that also affects how the territory are formed. But then the other parameter is the active scent time, which is a TA. You will see a TA through the slides as we go along. So now, just to give you an idea of the territory dynamics, so to understand that the territory at a given time t is a set of all the locations where the scent is active from any of the animal. Or if you want to think about, it's like for a given animal is where an animal has wandered within the last t minus ta time steps. Okay. So now if I'll show you a couple of movies. So this is just a still image. So this is a classical output for a model. Then I'm not showing where the animals are here, just the active scent. And then there are different colors for different animals. This is not such a high density. And you can see it's not such a high density because you see lots of white spaces. It means that within the last TA time steps, no animal has been there. And you see the form of this territory is not, a very ordered form, because the density is not that. So in fact, the active scent times control.
Your sound went down again.
Sorry?
Your sound went down again.
Oh, I see. Is that okay? How about now?
Now it's fine. Yeah.
Okay. Sometimes it goes. Oh, I see. Okay.
Yeah.
Well, tell me when it does, so I just repeat.
Yeah. Yeah. Sure. Thanks.
So let me just show you a couple of movies. So as you can imagine, the density control how dense the system is, obviously, but also how long this active scent time is. If you have a very long active scent time, then those scent marks remain on the terrain for a long time, so the movement is reduced, obviously. In fact, I want to show you, just keeping the same density, if I show you two movies, this is a fast territory dynamics. So if you just pick a color, you see that it goes pretty much all over, the space. There are periodic boundary conditions, and so they move very quickly around the entire region. And so this is probably not the real biological scenario. So let me stop this one. And instead, this one is slower. So, and look at this, as you can see here, more or less everybody after sometimes there was some initial condition that was put in, and you will see that each territory has sort of 6 neighbors, not necessarily, but this is probably a more biological relevant scenario. And you see the boundaries keep oscillating, fluctuating, and you see once in a while you see, this is the frames are I think it was every 2 times TAA steps. So, and you see the movement because of that. Otherwise, you wouldn't see much movement, obviously. So this is just to give you an idea of what the output of the model is. Now, how do we study this? Yes, so this is just because I wanted to mention the difference between a home range and a territory. So the territory is, therefore, it's a definition which is at each moment in time is indeed where the active cent is. Now, typically the data that biologists have is not about the centered territory, also it's very difficult to detect it. It's about where animals have been. And so, of course, this is what you might define the home range. So this is an area where animals tend to be over a certain amount of time. So you have to define the amount of time which you measure where it goes, and then you accumulate where it has been, and then you create a distribution if you want. And so out of one of those 2 movies, I forgot which now, I think it was the first one, sorry, the second one, then you can look at over a certain amount of time, what are these home ranges out of that model. And so you get plots like this, where now you see there is the X that you see there is where the animal started, well, it's where I started recording where the animal have been. And then this is the classical home. So each of these is a home range. And in fact, you have overlap now of the neighboring home range. The big one are next to the small one also. So there is a sort of very heterogeneous population distribution there. So these are the home ranges, and it's not the territory. Once again, the territory is at a given moment in time. So this is something integrated over time. So I wanted to show you this just to show you and give you an idea that all the heterogeneity that you see here is actually generated only dynamically. There was no disorder, because I'll tell you later at the end that, I haven't included any foraging process. There is no food. There always food is plenty available in this model. So that's why. So the heterogeneity is very much endogenously generated. Okay, let me come back then to analyze now the model. And so what are the parameters that we said were very important? So it's the density and the active cent time. And let's think about just the random walkers now. So let's not change the movement statistics. And what these parameters said, what I call the spatial competition parameter, is the ratio. It turns out to be, you can think about the 2 spatial scale, which is the space that an animal occupies in the absence of the others, and the average space available. So the average space available is what you expect given the density. And then the average space an animal occupies is the coverage, of the space, or if you want, the number of new locations you visit over time. So in fact, more precisely, you can call it that Z is a ratio with the number of distinct sites that a single animal visits within that time TA, which is the one that defines the area of the territory, divided by the specific volume. So, so these are the symbols I've used. So SN, and if you're using continuous time, N is the number of steps. That's fine. Otherwise, if this discrete time, it's just TA. And so, if you're interested in 2D to look at, some of the approximation we have used, which are slightly different from the Montreal approximation back in the 70s. So we sort of changed, probably improved the kind of estimate, and that's the one we've used because we needed a dependence on N to study this. So now all that matters, except when you're very high density or very low density, is that the spatial thermodynamics of the system depends really only on Z. And then as we go along, I hope I convince you about it. So let's see what happens now as we look at the dynamics of the function of this value of Z.
Can't hear you.
Yeah, can't hear you.
What about now?
No.
Can't hear.
Okay.
Yeah, now better.
Better?
Yeah.
So I said, let's try to look at the dynamics as a function of this parameter Z, the spatial competition parameter. So once again, imagine that you have the Z represent the pressure about an individual towards the neighbors, okay? So now let's look at the animals first. And we just look at the mean square displacement. And this is, I mean, I've plotted T as in relation to the active center time. And well, I just put a log scale. And what you can see, depending on the value of Z, when Z becomes very large, that mean square displacement essentially is nearly flat or flat. I mean, it's not picked up to go up higher anymore. And then if Z is much slower, so think about very low density or very short active time, this is like the first movie, if you want, everything moving so fast, then the movement of these animals is, there is no spatial exclusion because they rarely encounter anybody. And so the MSD is just completely linear. Now the saturation you see at the top is just because, you have a periodic boundary condition. So you have a specific value saturated, which is L squared over 6 in 2D, and L is the domain size. And on the other hand, once again, you see it's a log scale. So there is a caging effect, which happens for an extremely long time. So now, the idea is that how do you actually, so this is an interesting result, and it means that we expect that somehow animals are partially confined for a certain period of time. Eventually, the fact that the MST then eventually start growing again is that eventually they come out of this locking that they have, this caging. But so we wanted to go further and understand even more in details. If that is what happens, change in Z, we wonder is there really a different phases of the overall system as we look at in this MSD plot, which you can see it's very much continuous. You can't really distinguish it from that. So that we had to coarse grain the system to understand that and to look at the centroid. So I just want to show you a movie. So this is the idea how we coarse grain, because actually really we actually look at this movie and then we understand how to do things. So now there is a value of Z which is sort of high density if you want. And then you will see that as I show this movie, I'll zoom out in space. So I'm coarse graining.
Yeah, volume went down again.
Let's see if I actually. What about now?
Yeah, it's better.
So now you see that as I show you this movie, I'll coarse grain in space, but also I'll take the frame you see at the top. There is one frame, one steps. You see that I'll increase the number of frames as I look at. So I coarse grain in time as well. No? So you see, I'm coarse graining in space now. So these are the different animals. I mean, it's an hexagonal lattice. It's fine. So now after sometimes that we have far enough, and we have waited long enough, then you see the number of steps for each frame that you see is actually getting larger and larger. And now, as you can see, all that matters at some point is just where the boundaries are. Pretty much that's what defines the situation. And then you can even think about, okay, so it's like having some disk of variable size that interact, pushing each other around. So that's what actually we thought. I mean, this is just a representation of our ideas there. And therefore we decided simply to look at, even before the dynamics of the moving boundaries, if you want, just looking at the structure of the system and looking at the centroid. So what we did is that we look at the spatial arrangement of this centroid. No? And so now if I give you the different value of Z, and you see it goes from a very small value at the bottom, which is very much look like a gas probably, and something which is a bit more dense, something even further dense up there on the right, and it's not quite a real solid arrangement, that one, but then you have Z11, you see it's nearly a solid. No? So just looking at the centroid. In fact, if you look at the pair correlation function you plotted for these different cases, what you get is that you get the situation where the bottom one really look like the case of a pair correlation function of a gas. The intermediate one are the one that probably look like a liquid. And then as you see, once you break remember this is radial function. So once you break the radial symmetry and you see the distortion of the peaks at the top curve is indeed that because you come into a solid phase, or at least you expect to have a solid phase. So I just plotted them by shifting from 0 up so that you can distinguish them. So this was an initial indication that clearly there are different phases in the system. And so how do we go about actually measuring precisely these phases? Well, so now you have to look into, first, create, look at the 2D centroid pair correlation. And so when you look at it in those 4 cases I showed you, you see the D, which is a solid one, it is indeed, now suddenly you have an hexagonal structure. So you're clearly into a solid phase. Now the A, that's your pair correlation, it's like, it really probably like a gas. The B are those peaks that indeed represent a liquid. Now the C is the one sort of intermediate, yet the radial curve, the pair correlation function GR didn't show much, but in C, you might have clues that it's something else, it's something in between. No? And it turns out there is something in between for those that study this system in 2D system, phase transition in 2D system. And in fact, there is the well-known Coarse-graining Nelson-Young study about 2D melting. And what happens is that instead of like in 3D that you go from a liquid to a solid as you reduce temperature, then you can actually go in 2D to a state which is called exotic, where you have the location of the other individuals is still 60 degrees, but what you have changed is the translational order. So I'll tell you a bit more. So it turns out that we also have that phase in the territorial system, and so that's what we try to study. So now, how do you go and study this? Well, you have to look at the location of one individual in terms of the angles relative to the others. So there are these are order parameters which have been studied in the past. So we just picked them up, and this is the boundary orientation of the parameters. So you see that if everybody is at 60 degrees, so if you have an hexagonal tessellation, that's one. And if you don't, then you're going to get a value which is smaller than 1. And then you're looking at the angle between some reference axis, if you want, and the other J nearest neighbor. Right. And if you look at those 4 scenario that I gave you, and you color code it accordingly, and to look at at this boundary orientation order parameter, this is the local one. So each particle, depending where the neighbors are, so it's at the angles where they are, then you can see that the system goes from A, which was a gas, to B, which was a liquid, to this C, which already I told you it's sort of, you're going to see it's an exotic phase, to the solid. Everything becomes ordered. But notice that the C and D, they're not very different from each other. So in fact, the exotic phase is missing the translational order, but not the orientational order. So these are the local one. Then if you sum over all the particles in the system, that's when you actually look at the phase transition per se, and you try to find the actual value when there is a phase transition. But the other order parameter, which is for the translational order, to distinguish C and D, because in this case they're not distinguishable, is the translational parameter. And again, so these are things that get picked up from the literature, and that the way it's a bit more sensitive to actually detect these. But as you can see now, C and D, so the exotic phase is not as ordered as the solid one, and the value of Z, corresponds to, I just remind you those values that I picked up to show you the difference. So now, how do you actually find out? So if you want to find out if it's continuous of sharp transition or first order, so to identify the phase transition and the actual value at which it happened, so we plotted this global translational bond orientational order and then so the orientational and translational order parameter, and you can see there is a sharp rise right about those the value of Z, which one is something around 8, so let's close to 9, and the other one is very close to 10. So that corresponds to, indeed going into the exotic phase from the liquid phase and going to the solid phase from the exotic. So what I plot on the right-hand side, the column, the 2 on the right is the variance. So it's the susceptibility. So it's a variance of the side tip side series. And now, so sorry, I just forgot to say that. So we didn't do a real scaling analysis here. We could not. It was in computation. We couldn't do it really. So what we look into, since associated with this KTHNY transition, we can talk also about the topological defects that form during this transition, form or appear. And so we look into that instead. And so just for those that haven't found about these topological defects, so these indeed are defects in a crystal you can think of. You know, solid and exotic phase, they can have different kind of defects. So, and you know, I just put it down. They're very similar in sound, disclination and dislocation. So I also get confused. And actually, I have to tell you, I've done it for myself. So as I explained to you, so the disclination is in our case, in our model, was that we have a territory, instead of having 6 neighbors, which, you know, if everybody would have 6 neighbors, it's a perfect crystal. You might have 5 or 7, or even 4, 8, and so on. But we only look at 5 and 7. And when you have a pair of disclination, you have a dislocation. This is so. The red dots here are the territory which has 5 neighbors. The green are the one that has 7. And so notice when you have a disclination of 5, 7 together, this is called dislocation. And when you have pair of dislocation, and what I want to show you on the right, which is, although it's a defect, because they're not all, not everybody has 6 neighbors, but what you can see is that those lines that I drawn there, it just say to, you know, there is no destruction of the translation order. So in fact, that's why it's still a solid, although you have some defects. You know, you might have a more pure solid if you have none of those. And then the exotic phase, instead, you can see that you don't have this pair of disclination, so you don't have dislocation, because look at the one in the middle, suddenly, you know, you follow the centroid, and suddenly, you know, you have a defect and you interrupt those lines. So, and this is what happened indeed, you know, when you go into the exotic phase, different from a solid phase. And so now you can look at those, just the 5 and 7 neighbors. I mean, that's what we did. And then you can see that in correspondence where you lose, you know, free disclination, and you can see that where the, you know, the triangle curve, the curve with the triangle, where that becomes nearly 0, then you have the maximum of the dislocation pairs, you know, because that's where you got the pairs. And so that's the entering into the exotic phase. And then later on, when that decays, is when you enter into the solid phase. So we sort of managed to, without doing this, you know, the proper scaling analysis, by looking at these topological defects, we sort of convince ourselves that it's like a KTHNY transition that happens also in the territorial random walk model. Okay, so just to recap so far, because that was analyzing the, you know, the transition and the whole collective business of the model, is that what we had you look at the animal dynamics, and yes, we could see that, you know, you have partial caging effect, but you know, in order to explain why you have this different caging, so we had to look at the coarse grain level and look at the centroid and sort of figure out that you actually have different phases, and therefore the animal in there, they have this different MSD depending on which phase you are in. Okay, so now clearly having a collective neural model that gives you some interesting results, the question is always hard to say how do I match with experimental data. Well, if you have to do, you know, full simulation and match experimental data, it becomes very hard. So we decided to look into some approximate model that would give us an idea or represent what we see in the collective movement model. And so that's what we did. Oh, sorry, I forgot. Just for my biologist friends, they always ask me what do we need to learn about all of this, no? So what do we know when someone is a gas phase, you know, exotic phase, and so on. So then I plot for them, and we plotted in the book, if you remember, Nitan. So that's actually what that represents. So the fractional territory defended, depending which phase you are in the model, that tells you, I mean, the type, you know, different species defend more or less fraction of their territories, and that would be a way to distinguish them. So, okay, so this is just for my biologist friend. So let me come back to what I was saying. So that's the analytic model or the approximate model. So how do you do an analytic approximation in this case? Well, we started very simple. So we started from looking at 1D, obviously, and now you can think about an infinite number of these animals. And what became clear is that at least when there is enough high density, all that matters is that you have individuals, and then also what matters is where the boundaries of this territory are, which actually depends on where the others have been, obviously. So it's a very, you know, complex system, but the The point is that why not why not bothering only about a couple of those individuals and their movement and their extremal properties of them, so wherever they go. So instead of having, you know, all the scent profile that they actually live there, imagine if we can actually approximate where is the location of these territory boundaries and where the animal is. So that was the general idea, no? So to sort of get an idea if that was possible, we indeed look at two animals on a ring, and look at the model, or the territorial model of these two animals on a ring, and then we try to see what the feature that comes out of it. And when we do that, so this is out of simulation. So what we look at the MSD of either of the two, if you want, and then you can see that there is a spatial exclusion now, because in 1D, I mean, that MSD, once you have spatial exclusion, grows as square root of t. So if you don't believe me, trust me, that is a square root of time, and the initial low value of time is linear in time. So until they start encountering, you know, the scent of the other individual. No? So and then this one is the MSD, but then if you look at the actual occupation probability, is that, I mean, it sort of reached a situation where it's nearly flat top, not quite, because remember, this is this has to do with the fact that we have a probability of retreat, which is binary, so it's actually present or absence. You don't have such a smooth transition between one and the other. But of course, you have to imagine that this is plotted at a specific moment in time, and then if you wait long enough, you know, they go all over the ring, no? So once again, this is you want to know where this animal have been over time. That's what you plot. You see it's this flat top distribution, and but then it depends, you know, how much time you wait, no? So so how do you actually model this? So is there a simple way of modeling this? So the idea is that given that the dynamics of the boundaries, as you can see, there is a square root of time there, is qualitatively different from the dynamics of the territory, so they're much slower. So one thing about an adiabatic approximation, no? That's the first thing. So, but of course you have to do a sort of mean field approach, because if you want to think about that you have one individual in interaction with the rest, then you have to sort of, you know, input some characteristics of the movement of this boundary, and that's what we did. So you can think and exploit still the adiabatic approximation. So what we did, we can think about the probability distribution of an animal at X and the boundaries at L, and we approximate that by so we sort of do this adiabatic approximation. So you have the P of X, we say is given by the walker at X at time t. So this is an individual animal, given, you know, the boundaries that are at some location L, and then we just say it's, you know, multiplied by the actual dynamics of the boundary, which is now it's independent. So that's the approximation. And how does that dynamics of the boundary can be constructed? Well, phenomenological, you say, well, I can just imagine that as you've seen those movies, you always have a sort of pressure to go towards an average area, which is the inverse of the population density, roughly. And so we can say, okay, so if 1 over L is the inverse of the population density, then I have a spring, again, it's phenomenological spring, that is going to push me always to go into a size which is 1 over L. At first, that's what it would be. And so that's how we build this you know, toy model. And I mean, if you want, in the details of the math, I mean, they're not that complex. You know, you have to construct dynamics for the left and right boundaries. You know, the Q is about the boundaries, and it's independent of the walker. And you have a classical Fokker-Planck, I mean, diffusion in 2D, and then you have a spring, and therefore you have, you know, a gamma, which is your phenomenological constants, and this is left. So right and left boundary, L2, and minus this L is the inverse of the population density. And now this phi of t, just because in looking at the dynamics of this, this is what we wanted eventually also for looking at the data. So what's the actual temporal dynamics? This is like having a time-dependent diffusion constant in this Fokker-Planck, and that's we had the freedom to choose what that would be. And then in our case, if you put, you know, what we saw the MSD, if you put a square root of time there, actually, the integral of that is the square root of time. That's how we get the actual results of the simulation in one. So again, you can talk.
Yeah. Okay.
Okay.
Yeah.
Better now sound?
Yeah.
And we can say that, I mean, we can solve this exactly because you can just look at the boundary separation distance, lambda, and the centroid, and then you can solve, I mean, you separate the two, you can actually solve it there. I'm not going to write the solution, but, and then the dynamics of the walker now, remember, this is WXT given L. So wherever the L are, whatever they are, L2, L1, so we have decoupled them. And so you can just simply look at the solution, which is this, you know, exponential in time with this infinite series. And so by having, you know, this exact solution, then we can look at, you remember that we saw this mean square displacement plot that actually was going linear and then, you know, square root of time. So, so what you do, you just take your P of X, L2, L1, T, and you look at the mean square displacement, overall possible value of L2, L1. Yeah, I forgot to say that, you know, the solution of the Q's, you have to impose a boundary because you don't want the right boundary to become the left boundary. Okay, so that I forgot to say that. But this is accounted for. And this P is the product of the W and the Q's, and you do that calculation. And then notice that you got, now you get a phi of T dependency. Yeah? That will determine how, you know, the mean square displacement will grow. And this indeed, if you put this one becomes square root of time, is that what you see in the simulation. And in fact, you know, when you plot it, considering there is spatial exclusion, this is, you know, your square root of time. And sorry, these are the boundaries. So we can look also at the boundaries, sorry. But this is the MSD that we plotted earlier for the simulation. And that's a flat top if you look at, you know, the probability distribution that we saw earlier. And so now, how good it is as a model for the simulation? Well, clearly, as I mentioned earlier, the density has to be high. So you need to be in a very high dense liquid, exotic or even solid phase for that to work. And now you compare, you see how valid that approximation is, and then you see TAS, this is the same as TA. As that becomes larger and larger, that becomes, you go more and more into, you know, the solid phase, I mean, the exotic and solid phase, and then you see there is a good match between the model and the simulation, and then you know, it fails miserably as, you know, at the axis and times becomes small, so the system goes more into a very low density liquid, you know, you start losing it. So at least, you know, for the case where there is high enough density, it seems a good approximation. And so what we did, then we look into, I mean, consider a two-dimensional system. And let me tell you about the Bristol foxes now in the 90s. So yeah, so Bristol has a large population of foxes. And if you walk around the city, you know, and if you are attentive enough, you're going to smell clearly foxes, and they live in gardens, and they go out at night. In the middle of the night, you see them, they have no problem, you know, so that's their land, okay? And they go in, you know, they stay in different gardens, so an individual flocks, because they live individually, except when they're mating, they live in more than one garden, and they don't have a specific central location, so they don't have a nest. They keep living in two, three gardens, and yes, and they are territorial, and they have no problem with food. They're scavengers, so in a city they can find all sorts of foods. In fact, you have to worry about how you put out your the trash outside, and so that's our, you know, system to study. And now it was great, not for the foxes, but for us, that in the 90s the foxes went from a very high population to suddenly in 6 months a very sufficiently low population. So we could actually look into studying their behavior as, you know, this episodic of mange, which is like a scabies disease, decimated them. And so that was, you know, like a natural experiment for us. And so the data were just telemetry data, so not very highly resolved in space, but I mean, that's what we could use. And what we were interested in extracting from the movement data in the two scenario, if the act is sent, because that clearly is a parameter of the model that it's nearly impossible even to measure, you know, from an experiment in a fox, with a fox. So what is the number of days? Because that's what biology believes. It's in terms of number of days the scent remains active. And it turns out we could do it. And so, but the way you do it, since, you know, just in a brief explanation how you do it, is that, so now we tried to make it circular, and it was, you know, horrible math. And if I were to show you the math of this 2D system, it's even, it would be nonsensical. But the circular one was even worse. So we decided to go with a 2D model, which you have the 2 springs now, like the one you saw in 1D. So, and then now K is, do you remember this phi of t function I was telling you about? That is the time-dependent phenomenological time dependence, how these boundaries move. And so that has a constant K in appropriate units, which we could measure from the simulation and relate that in the simulation to the axis and time, and then use the model, which has these schematics on the left that represent that 2D model, the approximate one, to extract, to go and from the data, fit some K value, and then go to the simulation and obtain axis and time. In fact, you can, we plotted the relation between, if you have a random walk and you have a normalized territory border movement, which is K normalized appropriately, and then if you have a value on the horizontal axis, you go to the right, you hit the line, and then you go down, you find the axis and time. Okay, so that's the way it worked. So just to show you that I do have a little let's see, the parameters should be here. Yeah, so there it is. So there were like various parameters that we put in, but what I want to show you is that there was a pre-mange, which is the penultimate column, and the post-mange, which is the last one. And you can see that, well, first the population density was, let me see, yeah, you can see from the average steroid width, which is the third line. On the right, you see the pre-mange 435 in meter. This was a linear distance. You can multiply and make it square. And the post-mange is, double of it, at least. And then apparently what was surprising to us is to extract, as you modify the model, you don't simply put random walker, you put correlated walker, you have a velocity you have to worry about. The animal seems to move faster in the post-mange. And so we try to interpret this because, given they have bigger territories, they probably need to cover, more quickly the area. So that's why it's nearly three times as big. And the axis and times, it went from being 5 days when it was pre-mange before the population decayed to 3.37, which, given the small error bars, is pretty significant. So apparently, this fox, probably what talking to the biology, they said, well, okay, the model tells you this, but most likely this is because we have some assumptions. They believe that probably foxes realized that everybody was dying around them, and so they were not waiting that much to encroach into the foreign territory, because probably there was nobody anymore. So, and so that's what at least this exercise made us think about. So I call it elasticity because apparently there was an old hypothesis in biology which was formulated like 60, 70 years ago about that territorial behavior. We should think about like springs, and after we published, it turns out that there was this paper, it was talking about some birds. And it turns out that without knowing, we made a model with springs. Okay, so that was interesting. That's why there is a title, Elasticity of Territorial Borders. So, so let me conclude. I think hopefully I'm on time for questions. Okay, so what I've showed you, I've showed you some stochastic simulation of interacting animals without assigning a specific point displace, which is a den, which is perfectly fine for the foxes in Bristol. And if you want, I can add without foraging worries, because there was scavenger, lots of food around. And so I show you that an animal movement model where they scent, where animals deposit scent, is mediates interaction. And out of that, you have emergence of macroscopic, distribution of territories. And so now the words that Newton's like so much, conspecific avoidance makes territories undergo an exclusion process, and and essentially the correlation that you build up, they never die out, and that's why you have all this structure that emerge. And studying the phase transition, it appears that the system goes through a continuous liquid exotic solid phase transition. And I told you a bit about the fit to experimental data on Bristol foxes with the help of an analytic model. Now, I just realized that I miss out the slides where I thank all my collaborators. So, but since the names are here, okay, let me just thank them by looking at all the papers here. Jonathan Potts was my PhD student. Stephen Harris is a biologist, which had all the data. We have learned so much about foxes and their behavior from him. So then I want to thank Rubinstein and Simon Levine when I studied the stigmergy. That was the 5th paper there. Then I also got involved in looking at, okay, I haven't told you about the epidemic spread with David Sanders at UNAM in Mexico and his students, I forgot his name, Paris Becker, and someone called Kenkre, probably Nitan, I assume, so I have to thank him, I guess. I don't know who is there here. And then Alejandro Hebum Robles, this was my PhD students. That's when we started the phase transition that I told you about. And then my latest student, Sirana Salvaharm, this is when we look at the defects more recently to confirm the analysis of the phase transition. And let me thank again Nitan for the wonderful opportunity to write the book together. Thank you, everybody.
Thank you. Let's give an applause. That was so entertaining. So let's open it up for questions.
This might be not a exactly question, but this is a fantastic talk. And I remember, I think it was about 2008, when I was a grad student of Dr. Sen. And Professor Concrete gave us a similar talk at UB about the animals' behavior that time. I think was held in a Franzek Hall in our conference room. So that's a fantastic talk. The question is, I want to just a small question. Have you ever done the comparison between animals' behavior and plants' behavior? Plants' behavior, and I learned something about the plants' behavior. There's some similarity of how to make territories from here without the legs.
Yeah. Yeah, unfortunately, I'm very ignorant about plants. I'm very ignorant about, so I won't be able to say much about it. I've heard that plants, of course, you're right, they interact by, in different ways. Some of them is through, roots, but also, by shedding something from the flowers and so on. So, but I think it's a very interesting topic to work on, but, I really am completely ignorant about it. I'm sorry, okay?
But it's a great question. Yeah,
yeah, because one point of view is like plants do have a genetic weapons of how to make territories. So that's why I was interested in that idea. Might be very interesting if we can combine these two behaviors together and make some comparison studies.
Yeah, that sounds like a lot of fun.
Yeah.
But thank you again.
No, thank you.
Interesting and fantastic talk.
Other questions?
I have a comment.
Please.
Next time, I guarantee you that if next time you do not write the word conspecifics, you will have no audio problem.
So, but did we put in the book conspecific, or you make me put it out?
Yeah. Yes.
So what about primates, Luca?
This was a very nice talk, Luca.
Thank you.
Yeah, it was a fantastic talk. Luca, tell me a little bit about primates, because we poked around with primates. I sent you our own paper. Yeah, and I was just thinking that there must be similar stuff going on there as well, right?
Yeah, I think that, let's say like this. So in primates, I think what becomes important in primates is like this hierarchy that get developed in terms of social status, no? So, they, even without being territorial, so, because, you can have a group of individuals, so they're all within a territory, if you think about it. Nobody is shedding any scent, but they develop there is a self-emerging leader, which is generally because the strongest and so on. And so I'm not going to, so, and this is, you know, the paper you sent me about fission and fusion, this is what indeed, it's interesting. So at the moment I can tell you secretly, so nobody can talk about it, is that I'm developing models, this is for robots, where you use territoriality and hierarchy construction socially.
I see.
Create this sort of structure. I'm not sure it's going to be a biological system. That's why I'm thinking about robots really as an application, but I'm combining precisely this hierarchy construction with the formation of this territory. But now of course, they're not sending this robot. I mean, it's not very convenient. I mean, instead of sending, they remember where others have been in their head. So instead of having a memory in the environment, they have in their head. So that's why so they are sort of, they are not stigmergy. It's like now suddenly you use the power of their CPU, in other words. But it's much more difficult because it's just...
Yeah. Yeah, Sriram, please.
Question, comment. I noticed that, see, in your model, finally, the effective interaction between two individuals is more or less like a potential energy, right? I mean, it's actually like having a pair potential. It's like a liquid. It's true that it's done by means of this scent, which has a finite lifetime. But if you coarse grain in time, in effect, it's like a collection of particles repelling each other. So are there features which are different from equilibrium physics? Because, or what would you need to do to your model to bring out features that were different from equilibrium physics?
Yeah, you're right. So at the moment, no, I didn't do anything. So I think that we were thinking about looking at precisely the, you know, when you saw the movie where there were this disc, no, that there were, you know, sort of. So that's where I think one would have to go in and look, you know, at that aspect. So and try to address it. But no, you're right. I have not done it. Yeah, it's absolutely right. Yeah.
Thanks.
I've got a couple of questions, if I may.
Yeah, please, Subbu, go ahead.
Fine, thank you. Yeah, Subbu Ramakrishnan, University of Minnesota, Duluth. So the first question is, could you please comment on your approach versus something like, you know, Keller-Segal model or reaction diffusion type of equation approach the same problem, where, you know, when you precisely think of chemotaxy, but then instead of attractant, you've got repellent chemotaxy, right? So then of course it looks like a Fokker-Planck equation as well. You've got diffusion, and kind of building on Sriram's comment there, do you think you'll be able to better take into account the actual dynamics in terms of, say, gradients or where the flock is attracted and so on? So overall, your comments on your approach versus a more classical Keller-Segal type of reaction diffusion equation. That's question number one, and I'll wait.
Yeah, so I mean, it's very nice that you asked me the question, because actually, if I have to tell you secretly what motivated me about getting involved in this, because there are models from the 90s and up until the early 2000s where reaction diffusion type of models were built precisely for this, which is indeed, so now it was not a collective movement model, obviously. So what they were looking at wolves, okay? So there's the behavior of wolves and coyotes. Now, there are various publications. This is, it's been done in such a way that you take two individuals, they have their own nest, so they have a potential around which, you know, they are attracted to, and then you couple precisely, so they're diffused, but then they also, you know, they have a scent as well, you know, so they're field. So in other words, you have a potential field which represents the scent, and then there are two individuals that move, and then you can look at the overlap and so on. So this has been actually used to study the movement of wolves and coyotes. And so what you have, I mean, it's not a collective movement model, obviously. So you have, depending where you look at the data, where these different wolf packs are, then you have you identify from the data some sort of location, which is the nest of this different one, and then you can look at the interaction of the different wolf packs and so on. So now, so the reason that I was involved into actually building a different model is because foxes don't have a nest. So if you have no nest, then how do you actually form these territories? Because, you know, how do you actually do this overlap? How do you find out what that? So that's not the right model. Now, now the next question you might ask after you hear me here say this is that, have you ever compared the two models? Yes, we did. There was a publication which, you know, it's one of the one in the list, and they are very different, extremely different. So they overlap, I mean, the way they overlap and so on. I mean, so let's say this, we try to get close to that model by putting a centroid for each of our animals. And still in doing that, they are very different the way they're built and so on. So okay, so that's.
I see. Great. Yeah. Thank you. Should I wait for my second question? Or
No, go ahead.
Okay, great. So, thank you. So the second question is actually your perception of non-Markovianity, if you will, and memory, right? Because usually when we think about non-Markovian processes, one thinks in terms of processes where there is memory, right? So in this particular case, when you talk about a non-Markovian dynamics, are we talking some kind of time-dependent damping related to, for instance, for how long it takes for this scent to evaporate or something of that sort? Or lack of memory, and also all the non-local phenomena that would follow from a non-Markovian evolution, right? Does that give you any concern?
No, so but wait, so let's try to be clear here. So I mean, the system is, if you just think about each site having a value of that parameter which represents the active scent and so on, I mean, that's how you do the simulation. It's all Markov. Now the description, I mean, can be, as soon as you approximate, then of course it becomes non-Markov. Okay, so that's what you're talking about. Yeah. So as we do the simulation, we didn't bother at all about this aspect. No, so in the sense, you know, we do our simulation and then we analyze the system. Now, maybe your question is about, so if you do the approximate model, how no? So of course I made it Markov, as you saw the equations probably. There was no, I mean, this. So the only, so I try to capture some aspect of anomalous dynamics through the phi of t function, but you know, let's say like this, to some extent I avoided the non-Markov description. It would have been very difficult. Okay, so I don't know if I've answered your question.
Yeah, thank you very much. And also just to note, we've been doing some work on swarm robotics using ant foraging models. So, you know, if you would be open to it, I'd like to be in touch with you, send you a couple of papers.
Oh,
yeah,
some
of the things you're talking about seems very interesting. Thank you.
No problem. Thanks. Please do send me. So I'm into robotics.
Yeah.
Now.
There's a question from Sami.
Pardon?
Yeah,
Okay.
hi.
Go ahead.
Very nice talk. I have a kind of a naive question. So you're talking about the migration patterns in animals and such. Is there some kind of quantifiable study that can be done with migration patterns in humans? Perhaps even in small scale scenarios. I'm thinking of, let's think about within a city, you know, concentrations of restaurants or transportation patterns or something that you might quantify and make statements along these lines.
Yeah, so interesting question. Yeah, I mean, actually, I don't really know. I mean, I know there are models, you know, I don't know, probably you've heard these gravity models in social sciences. So probably you're more, you know, much more than me. So this is about migratory fluxes. In terms of the more micro level you're describing, I'm actually not sure really. I mean, there is a lot of literature as far as I know, but I'm not sure what's the flavor of the model. I think there are lots of simulations as far as I understand. Okay, so, but I might be wrong. So.
Yeah, that's.
May I ask a question, Luca?
You are allowed, yeah.
So this question that I have is about the previous question that was asked of you about the comparison of reaction diffusion and your model and your. So I was a little bit confused about that because that sounded like a nice question. You answered it in a way, but my confusion comes from the fact that to me a reaction diffusion process means a sequential process when there is motion and there is an event. Your model, I think, is wonderful. Your wonderful model, in fact, has something happening all the time. There is the walk. The walker is interacting with the boundaries, and the boundaries are interacting also. So to me, somehow, it is very difficult for me to put your model, which I think is wonderful, in the framework of a reaction diffusion. And therefore, to compare the two and to say that, one, whatever, better, this confuses me completely. So it confuses me out of the water, so to say. And therefore, I'm sitting down wondering exactly what is happening.
Yeah, so
my question to you would be, do you agree that these models, your model and the reaction diffusion model, are tremendously different from in concept?
Yeah, they are. And in fact, to some extent, you can think about that all these are event driven, because as soon as you encounter a scent, you actually, that's the event of the encounter that determines what you are going to do, specifically. And that's why we have this barrier that becomes important to model it rather than having, a field. Okay. So that's, I go along with you, what you said. Yes, they are very different.
It also seems to me that the overriding characteristic of your model is to replace the many body picture that we have got by something that is so much simpler. So there is a reduction against something that I do not understand as a characteristic of a reaction diffusion process.
Yeah.
And so this is just comments that came up in my mind for whatever they are worth. So yeah.
Thanks. Yeah.
I also wanted to ask about something that, so climate change. Basically, all these, climate change is playing havoc in animal behavior in many parts of the world, right? So my point is that can you, it seems like it should be quite possible to find relatively straightforward ways of building in possible effects of climate change in animal social behavior and modeling that.
Yeah, I think, yes. The effects, so probably the models that, gets as close as possible to detect this effect of climate is about migrations. So, because, you can study the migratory routes, the moment they actually take off to migrate and so on. And this, has been going on for some time. In terms of, these kind of models, which are much more local, if you want, at the moment, it's hard to consider in that context, but certainly there is a lot of interest in that respect. How successful they are at extracting the actual information from the characteristics of the changing climate, I don't know. But yes, there is certainly lots of interest, yeah.
One issue that the reason why I thought of climate change is because of food and rainfall. These two things are, very significantly affected nowadays. Not everywhere, but in certain